We're going to talk about modeling now. So in this case, you're applying a list of altered genes, proteins, and RNAs to biological pathways, but instead of looking at it at the network base as a uniform network, you're actually taking into account the fact that some of these, that genes and proteins and RNAs are different. They interact with each other in very different ways. And there's complex biology underlying these abstract networks. They attempt to integrate those relationships together in order to yield lists of altered pathway-based activities. And pathway modeling is really an arm of systems biology. There are multiple types of pathway-based modeling, and each one is suited for a different particular type of problem. So if what you're working on is a metabolomics and you're primarily interested in small molecule metabolism, then we go back 60 years and we're working with systems of partial differential equations, which now, because of large compute capacity, you can actually do computations across hundreds of interacting molecules. So this is not my field, but people seem to use CellNet Analyzer a lot. Then specifically for kinase cascades and signaling, particularly used in the drug industry, there are network flow models. An example is called NetForest, which are models based on kinase cascades, and given binding constants and KMs, will predict what happens when you phosphorylate a particular protein, what else will be phosphorylated, how will activities be affected. For transcriptional networks, there are a series of transcriptional regulatory models that have been built up from large RNA-seq data sets. The most common one in use is from the Califano lab at Columbia University. It's called Arachne. And then there are two more general types of modeling systems. There are logic graphs and probabilistic graph models, also known as PGMs, which take a pathway and then kind of capture the logic of up and down regulation interactions and attempt to infer what will happen down here when you affect something up there. And the advantage of these is that you don't need binding constants and rate constants. You're not actually modeling the physical chemistry. You're treating the thing as a flow diagram in a programming language. And then over the last year, there have been generative AI models which threaten to, and I hope will, upend this whole system and will replace these specialized modeling systems with more general and accessible ones. So I'm going to talk about a couple examples here, just to give you a flavor. So we'll talk about the Boolean models. So in a Boolean models, essentially you start with a detailed pathway description with all the biological relationships, and you convert this into a model in which there are two types of connections between the nodes, the genes. We'll say they're genes. Can have positive relationships and negative relationships, and then there is math, which deals with what happens when a positive and a negative relationship come into the same node. Who wins out? And then there's simulation software, which will allow you to do what-if experiments. Okay, is it not moving forward? There it is. Okay. Then in the last year, generative AI has really changed the paradigm of how people work, and uh, uh, have allowed people to apply the technologies used in chatbots to the problem of pathway inference. And so let me explain how this works. So the large language models like GPT that we're familiar with were trained to do a very simple task. They're trained to predict masked text. So the way these models are trained is you take all the text from the internet, you break it into chunks, you randomly mask out a word inside the text, and then you train the system with the masked text and the unmasked text to predict what was under the black bar. And somehow magically it learns all the relationships among words in all the languages of the world and can start generating what sounds like intelligent text. Well, you can do exactly the same thing, but with gene networks. So you start with a large set of RNA sequencing experiments, and people particularly like to use single-cell RNA-seq because you've got lots and lots and lots of data there. And you apply a perturbation. So you use a CRISPR inhibition or activation to change the cell state. Use single-cell sequencing to record genes that go up, go down, or don't change. And then you present this data set to a large language model and you mask out one of the genes and you ask it to predict it. And over time, after training, you end up with a system that's able to recognize all the myriad cell states that were in the data set and let you do those what-if experiments. What if I perturb a gene that wasn't in the original training set? Can I predict the output? Over the last few months, a bunch of these have come out. One of them is, most recent is SCGPT for single cell GPT. Came out from Bo Wang's group at UHN. And this was trained on single cell RNA-seq from 33 million cells, so a fairly large data set. This describes the training system. When they started with single cell data, they did the masking, and then they did a series to learn the basic cell states, and then they did a series of fine tunes to train it to do specific tasks like cell type identification, perturbation prediction, batch correction. And what they ended up with is, so here is just a UMAP of all the cell types that the system is able to identify. So it identified major cell types, intestinal epithelial cells, as well as a number of subtypes that people had not observed before because there were subtle patterns. So this is sort of an incredible resource of cell types here that needs to be explored. And then when applied to the perturbation prediction, what we're seeing here are they looked at a series of CRISPR experiments, 2 CRISPR experiments in this case, in which they knocked down 2 different genes. And they're seeing, you're looking at pairs between the observed change in other genes versus the predicted change. And you can see that the system is actually quite good at predicting how a gene will change. So in both cases, you can use either a Boolean model or you can use one of the AI models to predict perturbations to identify cell states. And now the question is, and this was top of my mind a couple of years ago, is all the work that we had done to curate the scientific literature and put it into human readable pathways, is that actually useful for anything? And a couple of years ago, my group did a study to ask the question of, do curated pathways actually allow you to make biological predictions? And so what we did at this point is ask how well you can predict the downstream effects of knocking a gene up or down, either by having experts gazing at pathway diagrams or using a inference algorithm. In this case, we used a Boolean network because this is before the AI tools had appeared. So I just want to take you through this. It's kind of fun. So the first thing we did, we went to the literature. We selected 10 cancer-related pathways. And we went to the literature and we collected from the literature experiments in which groups had knocked up or knocked down a gene in the pathway and then done an assay downstream to look at the pathway activity. And we weren't looking at RNA in this case. We were actually looking at protein-based enzymatic activities or process activities, because it's one thing to predict RNA expression. It's another thing to actually predict how the cell's going to respond. And we collected 4,968 pairs of upstream interventions and downstream responses. Okay. And then we did curation of 531 papers and found 847 well-tested cases which had replicated. So we have a gold standard set of perturbations. Okay, and then in a blind fashion, we did two experiments. One was we had reactome curators gaze at the pathway diagrams and try to figure out from what happened, what we would predict the target gene would do. And we also applied a Boolean inference algorithm, one called MP Biopath, developed in my lab, to predict the effect of the perturbation. And then the next step is, given those predictions from the curators and the empirical results from the scientific literature, what was our success rate? So the good news is it's better than random. The predictive accuracy of the curators was 81%. So 81% of the time, if they said a gene went up, it went up. The algorithm did almost as well. It did 75%. And in fact, it wasn't a statistically significant difference. And random guessing gives you 33%. When we looked in more detail at what the source of the errors was, it was primarily false negatives. That is, the curator predicted that, or the algorithm would predict that the target gene would not change, but empirically it did change. And when we dug into these cases, the vast majority of these were because there was something missing in the curated pathway. We had missed a gene or an arc or a reaction. So the good news is that if they predicted a change, they were very likely to be correct. That was up in the 90% range. And of course, what the implication of this is that Reactome needs more grant funding so that we can curate the rest of the biology and bring us up higher. There was also a very nice concordance between what the algorithm predicted and what the human predicted. It's an area under the curve of about 0.91, which is really good. And so since then, we have gone and made Boolean models for each of our pathways, and we're looking to get that published and apply it now. Okay, now with the generative AI models, we really need to do the whole thing over again using one of those models to compare. So the kind of in summary, pathway analysis allows you to discover the biological processes hidden in large-scale data sets. You've got lots of databases and tools to choose from. I've only given you a small sampling of them. The curated pathway database are actually worthwhile, and they've reached levels of completeness that allow you to accurately predict perturbations, and it's a very ripe field for machine learning. So at the end, I've given you URLs and other resources.